Dr. Atchison here, and we are talking about Chapter 6, the brain, physical growth, and health. And this first video is going to be all on brain development. So why should developmentalists be interested in the brain? This is developmental psychology. This isn't neuroscience. So why do we even care? Well, understanding the brain helps us understand behavioral development. If we understand the mechanisms that underlie these behavioral changes, we can really understand them a lot better. It also really helps us with an improved understanding of disorders. So we can understand what these disorders are. We can understand how to better treat them if we understand the neurobiology that's happening underneath. It's also important for intervention. As we'll talk about, there's a lot of critical periods and there's a lot of parts of brain development that are happening very, very quickly in infancy and throughout childhood and adolescence. And so being able to approach these things and understand how the brain is doing it in these different time periods can allow for earlier intervention. And we see that earlier intervention really has great outcomes, a lot better outcomes than later intervention. And again, it really helps us improve our theories of development as a whole. It helps us understand development of the whole organism, including the underlying effects that the brain has on that organism, the child or the adolescent. Okay, so here are the four main lobes of the brain. Stick your hands on the back of your head like you're relaxing and stretching. Your hands are on your occipital lobe. Your occipital lobe is primarily used for vision. Put your hands over your ears. Your brain, you're touching your temporal lobes. Those are easy to remember because it's kind of behind your temples, so temporal lobe. If you put your hands on the top back of your head, that's on your parietal lobe, and your parietal lobe does body sensations. Your temporal lobe, as you would guess if your hands are on your ears, does hearing. And your frontal lobe is kind of behind your forehead. And this is where we do a lot of the higher level kind of executive functioning. And what we'll see is that the frontal lobe is actually the last section of the brain to finish developing. We won't see kind of completion and organization of these synapses at an adult-like level until early 20s, early to mid-20s. So we see kind of brain development going on through a large part of the lifespan, and we see changes in these different areas. So the brainstem is the kind of bottom of the brain, and it includes the cerebellum, which is really important for movement and coordination, and it was the first part of the brain to develop evolutionarily. So if you look at our evolutionary past, and if you look at other species that are more primitive, you'll see that they will have a brain stem. They will typically also have a midbrain. Midbrain kind of went next. This serves as kind of a relay station, and it has things like the thalamus and the amygdala and all of that kind of are in our midbrain. And then last, we have our cerebrum. And that's when I was talking about those four lobes of the brain, I was talking about our cerebrum. And the cerebrum also developed kind of from the bottom and the back forward. So again, the frontal lobe is the last of the four lobes to really develop evolutionarily. So it's not a good psychology class if we don't have a picture of a neuron. You should be familiar with this. I know I drew a neuron in every single psych class I ever took. So it's a rite of passage. So it's your turn. Babies are born with about 100 billion nerve cells. So they have a lot of nerve cells when they are born. We've already talked about some of this brain development prenatally. The dendrite, if you look in this picture here, the little on the top of the picture, you can see where it's labeled dendrite. And that's the place where we get the information in from other neurons. So a neuron by itself doesn't do you any good. Your neurons need to be connected to other neurons. And so the information goes into a specific neuron through the dendrite. This information then goes into the cell body, and the cell body has to decide whether it's going to fire or not in the all or none process. Axons, here you can see it's kind of this tealish color. it's actually the purple underneath the teal, but this is what carries the impulse away from the cell body. This is what's going to take it to another neuron, okay? And here we see this axon is covered with myelin, this fatty sheath. And that's that teal, those little teal globules. What that actually does is it speeds it up. It speeds up the processing of the neuron. And the terminal buttons on the end of that axon are what communicate with the next neuron. Remember, a neuron by itself doesn't do you any good, and even two by themselves don't do you any good. You need a whole bunch for this system to really work. And so again, it's going to be connected to another neuron, and they're actually not connected. They actually have a space between them, and this space is called the synapse. And synapses we'll talk about are really, really important because, again, a neuron by itself doesn't do you any good. You want this neuron connected to at least one other neuron, preferably more neurons. And so we'll talk about these synapses, these connections, being really, really important. And we'll talk about how they're formed, how we can get rid of ones that aren't useful, and how all of this is an ongoing part of brain development. So the stages in neural development, we start with proliferation, which is really the formation of neurons. And again, most of this is happening prenatally. So most of our neurons are formed between 10 and 27 weeks prenatal, and they are going really, really fast. So 250,000 per minute is how many neurons are being produced kind of in this period. So we make all these neurons, then we have migration. Migration is where those neurons have to actually move to where they're supposed to be in the brain. So these are going to actually migrate, and they actually have to move to different parts of the brain, different layers of the brain to organize themselves. This is typically done by about 7 months prenatal. We're not really sure how this works. There's some evidence for fate maps, like these neurons kind of are fated to go to certain places. There's some evidence for chemical attraction, that they are chemically attracted to different locations. And there's some support for these are being supported by glial cells, which is, again, what those myelin sheath is made up for. When we talked about teratogens, we talked about how bad alcohol was for brain development. One of the reasons it's so bad is because alcohol is affecting both peripheral proliferation, how many neurons we're making, but it's also affecting migration, where those neurons are going. And both of those are really, really critical building blocks in the brain. So we get all this stuff done by about 7 months, and then really what happens from about 7 months prenatal to about 25 is we have elaboration or differentiation. This is when these cells are growing, these cells are myelinating, these cells are making connections. That's what synaptogenesis is. Synaptogenesis is just a fancy word of creating synapses, and again, making those connections is going to be very, very important. So synaptogenesis is, again, just neuronal connections. We see this is really, really immature, about 4 months gestation, but we start to get some mature neurons by about 8 months. And this is really, really rapid in the first 2 years of life. So we're really doing a lot of brain growth in the first 2 years. If you think about the difference between a newborn and a 2-year-old, a lot of that has to do with synaptogenesis, organization and connectivity in the brain. And we see that this will, again, continue out throughout the life, including, again, all the way up until our mid-20s. And we see peaks in different brain regions at different times. So again, that frontal lobe, really, we're not finishing that until our mid-20s, but we'll see a burst in the visual cortex at about 3 to 4 months. And we'll see but a peak between 4 and 12 months. So again, we see these differences. We see a peak in prefrontal cortex connectivity happening at about 2 years. And again, that's that executive function in the frontal cortex. The other thing that the brain does is it going to make more synapses than we need, with the idea that more is more. It's going to overproduce them. It does it this way so that we are available to adapt to our environment. We talked about nature and nurture. So nature says, okay, well, we're going to give you a set of connections that will work whatever your nurture is, whatever your environment is. So we see this really overproduction of synapses and making connections that maybe we don't ultimately will need, but we're going to start with those connections and then we can get rid of them later. And that we do get rid of them, and that occurs in different areas and in different time periods. So we see the visual cortex is really, we're kind of eliminating these primarily around 6. And again, that prefrontal cortex, at the very, very front of the frontal cortex, is that last to finish, and it doesn't really even start till mid-teens. And so again, we'll see kind of different areas of this. So when I say that the brain's not done until 20s, what I'm saying is all the connections that you're gonna have are pretty much there. But what's happening is we're getting rid of the ones that aren't useful, and we're strengthening the ones that are. So this is that kind of that idea of undevelopment too. So we not only kind of create more synapses than we need, but we also create more cells than we need. And so some neurons die. When we say that brain cell died, brain cells die. And this can be very adaptive too. This is not necessarily just your brain on drugs kind of situation. We're actually, there's some benefit to selective cell death when it's happening naturally and in a healthy situation. And those neurons that don't have good connections, those neurons that aren't being used well, that aren't being utilized, that aren't really effective, will die. And that allows more resources for those other neurons that are working, that do have good connections, that are really being utilized in a very adaptive way, they'll have access to more resources. Again, synaptic pruning is also happening in this undevelopment, where we're getting rid of some of those synapses. So it's just what it sounds like. If you're pruning a bush, you're cutting away the things that it doesn't need. You're cutting away the dead stuff. You're cutting away that's stuffed so that it can grow healthier, right? The same thing happens in your brain. Connections between neurons will be pruned away, will be cut away, be gotten rid of, so that the strong connections that are really adaptive can be stronger and can be more utilized. An example of this is we see this is really happening in the primary visual cortex by the end of the first year. So some of those big changes that we see in vision are coming from this organization, this undevelopment in the primary cortex at the end of the first year. So myelination is, again, those glial cells, again, in that picture, those were those kind of teal little kind of globules that were on that purple axon. And it's a fatty sheath on the axon, and it really, again, speeds up processing. So it makes, instead of that neuron, kind of just continually going, it's kind of like a booster pack, every glial cell it hits. And it boosts that speed up. And it allows for quicker transmission, which is, of course, very adaptive. We see that this myelination is something else that's happening in brain development over the course of infancy, childhood, and adolescence. We see that in the sensory cortex, it begins before the child is born, and it continues through that first year of life. But we see that these associations, these cognitive, the association cortex, where different parts of the brain are talking to each other, is again occurring into adulthood. So again, this is another one of those things where the brain's not finished yet until mid-20s, where we're strengthening these connections between the different areas of the cortex with these association areas, and we're speeding up the processing of them as well with myelination. And again, this is really affecting that speed of the processing because it's making those impulses down the axon travel a lot faster. So a newborn's neurons are 16 times less efficient than adults' neurons because they don't have the same level of myelination that we as adults have. And we know that myelination is really influenced by specific experiences. We can see individuals that are deprived of light have decreased myelin production. And we can also, there's been studies with rats where they've given increased activity, given them more opportunities to be active, and we've seen increased myelinization. So again, these are one of those things where nature is definitely playing a big role, but so is environment, so is nurture as well. So here are some milestones in brain development. You can see, look across the axis at the bottom, and we see that birth is going to be at 0. And that when we have a negative number, we're talking about kind of prenatal development. And again, here you can see that's where that production, that proliferation, and those migration of neurons are happening prenatally. And again, that's done about 7 months. And you can see that's where we start myelinization then. Once the neurons are to where they're supposed to be, those yellow lines start myelination. And that continues again into adolescence and into adulthood for certain areas. And again, we'll see different areas of the brain kind of peaking at different areas. So the green line is the auditory and visual areas of the cerebral cortex. So we're talking about areas in the temporal lobe, areas in the occipital lobe. And we'll see that those really kind of get done first. We see a really kind of a peak in about 6 months, 5 to 6 months. But those areas are pretty much done at around 6 years of age. Okay, whereas the blue area is going to be language areas of the cortex. We see a really big peak in that around 10 months of age. And again, we're also done to about 6 or 7 years. When we talk about language development, we'll see some of the kind of outcomes of this. So if you speak a foreign language, if you speak more than one language, if you're bilingual, or trilingual, if you speak with an accent, chances are you didn't learn that second language until after 6 or 7. And if you do not speak either language with an accent, that's because you got that information before 6 or 7. That critical period of language development has to do with this brain development. So again, this exposure to a second language before 6 or 7 really changes how the brain processes this language. And the brain says, oh, well, I need to keep more of these connections. I need to keep more of these cells. I need to keep more of these abilities to understand these 2 languages because I'm using them. Whereas if we're only being presented with one language, I am definitely a single language speaker, especially before the age of 6 or 7. My brain didn't need that. My environment didn't call for a multi-language environment. And so my brain will have gotten rid of the ability to tell the difference between those different phonemes. We'll have gotten rid of some of those abilities that's happening here in this area of brain development. And again, the frontal cortex, this purple line, the frontal lobe is our last area to really finish processing. And for all of these arcs, what those are talking about is rapid synapse formation, so that connecting those 2 neurons together. And then again, pruning back the ones we don't need. So that's a lot of what's happening. And again, we don't typically see the adult level of synapses really until about 15. And then we again will have continuation of myelinization and different other kinds of brain development, so that our prefrontal cortex really isn't done until our early to mid 20s. So we have a lot more ability to understand the brain and know about the brain with the creation of different techniques to allow us to image the brain. One of which is PET scanning. And PET scanning, what we do with that is the individual will ingest a very low level of a radioactive substance, and then it's used to measure metabolic activity in the brain. So we can look at activity based on how much energy different parts of the brain need. And so that's what we'll do with PET scans. PET scans give us pretty good scans in terms of quality of what we can see where it is. PET scans are not good on timing. We don't get good information on how quick processing is happening because of that kind of radioactive isotope and how it's processed. Our best pictures really come from fMRIs, though, and this is using a magnet to record changes in the oxygen levels in the brain. And again, our best images are coming from fMRI, and you'll see a lot of research now is done with fMRI. The problem with fMRI, there's two. One's a practical one. It is crazy expensive. And so you have to have a lot of money to do an fMRI study. There's also another practical problem of doing fMRI research with children. To do an fMRI, you need to be still. Good luck telling a small child to be still. And so to do those studies with small children, they actually kind of bind them. They like very tightly swaddle them so that they can't move, which of course small children love. So we don't see as many of these kinds of studies done with small children, especially infants. What we tend to see more of with them is ERPs. The other problem with fMRIs is that fMRIs don't give you, again, great temporal information. We don't know about how quickly things are being processed. Whereas with ERPs, we get really great temporal information. We can really understand and see activity very, very quickly, and we can see changes in activity very, very quickly. ERPs are done with an EEG machine, which is kind of a cap that they put on the brain, on the skull, and it measures the electrical activity in the brain. But the problem with that is those electrodes where it's measuring that is basically taking a core sample. It's saying, okay, there's electrical activity kind of in this range of the brain, but we don't have a lot of great information as to where particularly that's happening. And so what you'll see is a lot of researchers will take information from ERP studies and information from fMRI studies and use those together to kind of develop theories because each of them has a benefit. ERPs are done a lot with kids because it's kind of like wearing a hat. And so a lot of kids are really okay with that. And so they'll do EEG and ERP studies with very small infants as well as children of all ages and adults. And again, this activity that we're measuring tells us about the excitability and organization of parts of the brain. What's really nice is, and that's the difference, is of an fMRI versus an MRI, is we can look at these with behavioral changes. So we want you to do this, and then we're going to measure what your brain does. And so we can really pair these things up and ask questions and get answers about different areas of the brain and about different areas of function, such as surges in the frontal lobe. In frontal lobe activity has we've seen are related to inhibition of behavior, saying, okay, no, I'm not going to do something. I'm not going to do a behavior. Planning, goal-directed behavior. And again, we also know that this is due to a production of synapses and myelination there. And again, we can see this with both EEG and fMRI. And again, EEG is the method in which we're using to look at ERPs. So those kind of go together. The two E's kind of go together. So you can remember that way. We can also, we can see that the experience plays a big role in brain development as well. So plasticity is the responsiveness of brain to experience. So again, we've talked about plasticity before, how plastic something is, how malleable it is, how able it is to change. And again, your brain is set up to kind of be ready for whatever environment it's going to be in. So it has a lot of plasticity. And we're going to talk about two main kinds of plasticity that your brain has. And this is work that was done by Greeno, Black, and Wallace. The first kind of plasticity we're going to talk about is experience-expectant growth. Okay? And this is the kind of plasticity that your brain expects to get. Okay, so for your brain to grow normally, it needs some of these really ordinary experiences, like seeing things that have borders. Well, pretty much everything around you has a border. So as long as you are getting input through your healthy eye into your healthy brain, that typical development will happen, again, by seeing these different things. And an important mechanism is synapse overproduction and elimination. So again, the brain is making more connections than it needs, and then it's pruning away some of those synapses, so we just have the most effective ones. So these experience-expectant growth things would be these kind of major things. So seeing and hearing, the fact that we're going to hear things, we're going to see things, we're going to touch things. For typical brain development, we need that. We need those kinds of experiences. And again, these are those kinds of plasticity that the brain expects to get, that every human should be presented to as long as everything is typically developing. And so again, we'll see that this allows the brain to grow in particular ways. The second one that we're going to talk about is also done by Greeno and Black, and this is experience-dependent growth. And this is the idea that the brain changes depending on that particular individual experience. So what language you learn depends on what language you are hearing around you, you are experiencing. Did you learn one language or did you learn more than one language? Again, that depends on the experiences that you're having. It's dependent on that experience. So your brain will develop differently if it's hearing more than one language than if it's hearing one language. And again, that is dependent on the experience. Other experiences can include music. So if your brain, if you are experiencing music and playing music and reading music, your brain will keep and develop according to that environment. Whereas if you don't, your brain will not develop according to that environment. Reading, writing, video games, these are all these kinds of things that are dependent on our particular environment, and to some extent dependent on the time period that we live too. So clearly brains 100 years ago didn't develop playing video games. So they had different plasticity. They grew different ways because of the different kinds of experiences that they were having. And so again, these experience-dependent growth is really focusing on these experiences and how those influence neural development. Other research that's done is non-enriched versus enriched environments. And so this was done with rats. And what they did is they had kind of two rat communities. They had a rat community that was like a really great place for rats to live, and it had lots of cool rat toys and lots of cool rat tunnels, and lots of great enriched rat environments. And then they had another group of rats, and those poor rats had the non-enriched environment, and they didn't have much to do at all. It was a very plain cage. They had their water, they had their food, and that was about it. And they sacrificed these animals and looked at their brains. And what they found is that the individuals that had the enriched environments, the rats that had the enriched environments, had heavier and thicker cortexes, they had larger neurons, and they had more dendrites, which meant more connections. And this was found for both young and old rats. What's really interesting is that they went back and they did another study looking at this, and they added a third group. Okay, so they had super rat environment with all this cool rat toys that are human-made rat toys, and then we had non-enriched environment, which was really boring. And they added a middle group, which was natural rat environment, and this was set up to really simulate a really natural kind of habitat that a rat would live in nature versus these lab environments. And they found that those rats had the best brains yet. That this really natural environment that was presenting them with things that they would encounter in nature gave them the best brain development. And this has to do with kind of the evolutionary history. Their brains were designed to develop in this natural rat habitat, not in super rat cage or really boring rat cage. And so we see differences in brain development depending on those interactions. The brain is not all doing this before 2. There's a lot of other stuff going on. So we see from 2 to 6, the brain size really goes from about 70% of the adult size at 2 to about 90% of the adult size at 6. And by 4 in the cerebral cortex, so again, those are those 4 lobes, that's where we have the overproduction, myelination, and cell death and synaptic pruning. So all of those things are going on. And we really see this improvement in preschooler skills. If you, again, compare a 2-year-old to a 6-year-old, which is a kindergartner or a first grader, they're night and day. And again, a lot of this has to do with those changes in the cerebral cortex. We see increased connectivity between different regions in the cortex, which is going to be really helpful for function. When those areas of the brain can conversate with each other and communicate with each other, we'll see better outcomes. We'll see changes in the cerebellum that are going to aid in balance and control of body. And that's going to help with a lot of the motor gains that we'll see in this time period as well. Again, there's a lot of brain development going on in adolescence too. In fact, there's a whole video that I want you to watch called The Teen Brain. It's about an hour, but it's really, really good. And so I encourage you to watch that. It is required. Because I only gave you one slide on this, so really watch that. We see that this pruning continues, especially in those frontal lobes. And so we'll see a lot of changes in adolescents' ability to pay attention, to plan. That means good decision making, those sorts of things. I like to blame all my bad decisions in high school and college on the fact that my frontal cortex wasn't done yet. Now, whether that's legitimate or not, I'll digress. But we again know that area of the brain is important for things like planning and decision making. We'll see that, again, we'll see increased growth and increased myelination, which will again speed up processing, and it will strengthen those connections between different areas of the brain. Sorry about that. And we'll also see changes in neurotransmitter responses. We see that adolescents' brains are more sensitive to these excitatory messages, which really intensifies reactions to stress. So the fact that a lot of teenagers, it seems like it's going to be the end of the world, has to do with how their brains are processing neurotransmitters. So some of those behavioral differences that we see in adolescents are really indicative of these brain changes. We also see that they're more sensitive to pleasure and novelty, which again explains a lot of kind of the behavior of adolescents. But because this frontal lobe's not done, they don't have the capacity to inhibit these impulses. We see that nutrition actually plays a really big role in brain development as well. The next video is almost exclusively about nutrition, so we'll talk about that a lot more detail. But we know that it really affects the cell number, the number of neurons, the size of those neurons, whether those neurons get to the correct places, the connections those neurons makes, and whether those neurons get myelinated or not. And we know that malnutrition during proliferation, which is again happening prenatally, really has permanent effects that cannot be undone. So nutrition is again a really important piece of that. We also know that again iodine is really important both prenatally and throughout development in proper brain development. So that's one of the reasons that their salt is iodized, so that we are making sure that our children are getting the amount of iodine they need for proper brain development. So this is just a recommended reading. I am addicted to this book. I love this book. It is really written for public, so it's not written for academics. She is an academic, but she's writing it for parents and families, and it's excellent. I learned things from that I didn't know before, and this is my area of expertise. So she does a great job of doing this. So it's the unexpected side effects of classrooms, ballparks, family rooms, and the minivan. So I really, if this is an area that you're interested in, especially if you're interested in development as a whole, this is really great. I recommend it to families and my friends, and they've all really, really liked it. So that ends our lecture for today, and I will see you in 6.2 lecture. Bye.